Hello children, how are you? I hope you all are enjoying this online class and working very hard and using your precious time in studying and covering the syllabus and following all the instructions given by your teachers. Okay, just to have a quick revision of what we have done in the previous class. We have done it started with the important components of a cell. There are three important components. One is the cell membrane, second one is the cytoplasm, third one is the nucleus. These three important components are there. So we started with the cell membrane and we studied that cell membrane is the outer membrane. It is also called as a plasma membrane and it is selectively permeable. It selective means it only allows some of the things to come inside the cell and some of the things only to go out of the cell. Okay. No, now, and we have also done like what are the components to form the cell membrane. So basically a cell does the transport of the components from the cell to outside and from the outside to the inside. So within the once intercellular transportation is done by the plasma membrane. So today we will start with the types of transport that is done by the plasma membrane. So basically there are two types of transport system done by the plasma membrane. One is active transport, another one is passive transport. Now from the name active, that means it is very fast, and passive means little slower. But here along with the fast and the slow, there is one more thing is added in the transport process. Active transport, that means if the substances has to pass through the membrane, but to pass or cross the membrane, they need some kind of energy. They need some kind of energy, then only they can jump inside or jump outside. So that type of transport system is called as active transport system. And when it does not need any energy, just pass across the membrane without the expenditure of any energy, this is called as passive transport. So just there are some definitions you will note down. What is transport? Substances may pass across the membrane without or with the expenditure of energy. So substances can be passed with the expenditure of energy. Substances can be passed without the expenditure of energy. So the one active, when the substances pass the membrane by consuming ATP, ATP is the energy currency. So when the things are passed with the help of ATP molecules, then it is called as active transport. And when the substances pass the membrane without consuming any ATP molecules, this is called as passive. This is fast, this is slow. Bulk transport. When large amount of substances or food particles pass across the plasma membrane, that means not one or two, lot of components are passed, that is called as bulk transport. And the process of bringing the bulk of materials from outside to inside by the formation of endosomes is called as endocytosis. Cyto term is for a cell. When the things are passed from outside to inside, this is called as endocytosis. When the bulk expulsion of material as a secretion or accretion is done from the cell, it is called as exocytosis. So, okay, you have understood this. So you must have copied this 1, 2, 3 and 4, 5 definitions: active transport, passive transport, bulk transport, endocytosis, exocytosis. Okay, now I go for the next topic. This is a kind of a picture diagrammatic representation for it. You can see that the active transport, which is done by the utilizing the ATP molecules, it can be done in the three ways. One is uniport, then it is symport, then it is antiport. These two are called as co-transport. That means when two substances are transported at the same time. So here at a time only one substance or one component is passed across the plasma membrane. It is called as uniport, and when it is done, two are done together, then it is called as co-transport. Co-transport are of two types. One is symport and one is antiport. Uniport means When any single component passes the plasma membrane from out to in or in to out with the use of ATP molecules, it is called as uniport transport. And in symport means together. When two components, by the shape of the component, you can make out this is one circular, this is one triangular. Two different substances are there. They both are following the same direction. Both are entering inside or both are exiting outside, maybe. So in that case, it is called as symport. Anti means one is coming inside and one is going outside, then it is called as antiport active transport. Okay. Now, we come to the passive transport system. Okay, the passive transport system, in this we will do two types of passive transport. One is the diffusion, other one is osmosis. Till now you might be aware of these two terms, diffusion and osmosis. Diffusion. Diffusion is what? Simple. The process of movement of the substances, solid, liquid, gas, any solid particles, liquid particles, gas particles, from the region of higher concentration to the region of lower concentration till a uniformity is maintained is called as diffusion. And when this diffusion takes place, there is no membrane is crossed. Now, you can see that in this picture, this is suppose A side and this is B side. Okay, so now there is no membrane. Here the concentration of this yellow particles is more as compared to this side. So what happens? So the movement of this yellow particles is always towards this side because the concentration is lower here and the concentration is higher here. Now you must have seen that if there is a ink, there is a kind of a, we just put the ink. What happens? When it is just one container, and if there is a ink in the lower bottom, what happens? The ink starts moving upside because what? The concentration of water is more this side and the water is less this side. Water is more concentrated in the bottom, water is less concentrated in the top. So the movement of water molecules does like this, and gradually what happens? The color also gets uniformly spread. Okay, so this is one, and in this there is no energy consumption is done to do this kind of transportation. So this is called as diffusion. Okay, so now what is the importance of diffusion? First one, it helps in the spread of different substances throughout the cytoplasm of the cell. So suppose if something gets inside the cell, so what happens? Gradually it starts, because suppose if something gets inside the cell, so definitely the concentration of that substance is low inside than only, like suppose we take the example of oxygen and carbon dioxide. When the cell completes the respiration, the CO2 content is more inside the cell and the CO2 content is less outside the cell. So what happens? The movement of the CO2 will be always from inside to outside. So what it will be? Exocytosis. But in the same condition, when the oxygen is already utilized for the respiration, so the oxygen concentration is low inside and the oxygen content concentration is high outside the cell. So what happens? The diffusion will take place from outside to inside. So it will uniformly spread in the cytoplasm of the cell because each organelle will get the uniform concentration of oxygen. So first one, it spreads uniformly in the cytoplasm. Okay, osmosis is also a type of diffusion, but here only solvent is allowed to diffuse. Now, one more thing how this diffusion rate increases. Diffusion rate increases with the temperature. Number one, temperature. I'll give you one quick example for it. Like you must have seen, when something is cooked at home, what happens? It is hot. So the smell of the food spreads very fast to the inner part of the house or maybe to the neighbor's house. Why? Because when the aroma of the food is hotter, the rate of diffusion also higher. Okay. Second, the diffusion rate is affected by the density. Like suppose the density of any component is more one side, so definitely the diffusion rate will be higher. If the density, where the density is more, what happens? It will move towards the other side where the less dense is there. So do remember, from always from the dense to the lower dense area, the diffusion process takes place. Okay. Next point is diffusion helps in exchange of respiratory gases between the cells and then environment. I have already told this to you. Transpiration is also a diffusion process. How? What is the meaning of transpiration here? Transpiration is the loss of what, like loss of water from the excess water from the plant through the leaves, through the stomata. Water vapor comes out and it condenses and forms the water droplets. So what happens? Inside the plant, the water is more, concentration of water is more. So what happens? It comes out of the cells of the plant. So that is also diffusion. Flowers spread aroma through the diffusion to attract insects or other animals for the pollination. Just now I gave an example of food. So what happens when the flower blooms completely? The aroma spreads. So more surface area, definitely the aroma spreads to the wider areas, so that the insects get that smell of the nectar and they come closer to the flower to do the pollination. Next is osmosis. I have already discussed that osmosis is also a type of diffusion, but there is a slight difference between the diffusion and the osmosis. Osmosis is only restricted to the solvent liquid components. So just read what is the definition. The diffusion of water or solvent across a semi-permeable membrane from a region of its higher concentration to the region of its lower concentration, that is called as osmosis. And plasma membrane acts as a semi-permeable membrane. Now you see this. This is a semi-permeable membrane. Now here, this always if the solute particles are more in one region, so definitely the osmosis, the concentration of the water is low in that side. That means this is highly concentrated, because this side, suppose this is simple water, this is the sugar solution. So sucrose molecule is dissolved because in between the water molecules, the sucrose particles have already taken place. Okay, so what happens? So if you will keep like this, is semi-permeable. That means it will allow only the solvent molecules to move towards. So what will happen? So water molecules will start moving this side, and some of the sucrose molecules will also start moving this side. But massive distribution will be done from this side to this side, because the water concentration is higher here and water concentration is lower here. So what will happen? The movement of the solvent will be this side, and some of the solute molecules will move this side. Okay, understood? Now, what is the difference between diffusion and osmosis? First point, you write down this, children. This is very important. First, it can take place in solid, liquid and gas, but it will only take place in liquid medium. It does not need any semi-permeable membrane. It needs a semi-permeable membrane. It equalizes the concentration of substances. That means, like from A to B, if it moves, so it stops only after when both the side equal number of molecules of the solid or liquid or gas maintain, then only it stops. But it does not equalize the concentration of solvent on the two sides of the membrane. No, it will not be equal. It can be 90, 80, 60, 50, like that. Different substances diffuse independent of the one another. That means if there are 2-3 things mixed with each other, so they will do according to their rate. Here only solvent undergoes diffusion. Like, okay, So like sugar solution, what was moved? Only water was moved. Diffusion is not influenced by the presence of the substances, but here in the case it is influenced by the concentration and the type of solute particles. Now, I don't know whether you know or not, there is an instrument called as osmometer. Osmometer we make of potato. Osmometer tells you or demonstrates you the osmosis process. Okay, how? We take a potato, big potato, we make a groove inside the potato. We slice into two half, and one half we take like this and we make a groove type of, so that we can fill something inside that groove. What do we fill? We fill the sugar solution in that groove of the potato, and then we place that potato with the groove in water. We keep it, we take a tray type of thing, and we keep that potato filled with the sugar solution in the water, and we mark that area in the groove till where the sugar solution has taken place. With the pin we just put it. After few hours we see that the water level inside the groove has crossed above the pin. What does it show? The water concentration was lower inside the groove because solute particles was more, and the water concentration outside the potato was more. So the where the osmosis done, from the outside environment to the inside of the groove, the movement was done. So this shows that the solvent, and what was the semi-permeable membrane? The potato layer. Okay, so this shows that only the solvent molecules started moving from outside to inside. There is no solute particles movement was seen. Now, osmosis rate and the condition of the cell depends upon the osmotic solution. There are three types of osmotic solution, okay, which will cause the osmosis. One is hypotonic, second one is isotonic, third is hypertonic. Hypotonic, which has an osmotic concentration lower. Hypotonic means in which the solute osmotic concentration means if what amount of solute is dissolved in it. So hypotonic means definitely the solute particles will be less as compared to the other solution. Hypo means it will have more water, less solute. Isotonic means almost equal, and hypertonic means when the solute particles are more in the water and less than the water. So what happens? Suppose if inside the cell water molecules is more and the solute particles solute concentration is less, and if it is kept in a hypertonic solution, that means where the concentration of solute is more, what will happen? Which side the direction of osmosis will take place? Osmosis will take place like this side. The definitely the movement of water will be from this side to this side. So what will happen? The cell will lose its water amount from inside to outside, so it will shrink. Okay, but in the isotonic, what will happen? Both the side movements will be done. There will be no change in the shape of the cell. But in case of this, if the cell is having less water, like the and the outside in the environment more water is there, that means environment is hypertonic and cell is hypertonic. So what will happen? From outside to inside, the water will move towards the cell. Cell will become turgid. Turgid means swell up. Turgid, and in the first case, the cell will become flaccid. That means it will shrink. Okay, so next is, and on the basis of the hypotonic and hypertonic solution, there are two types of osmosis. One is the endoosmosis, one is the exoosmosis. Till now, you must be aware of the endo and exo. Endo means when the water is entering the cell from the outside environment. This is called as endoosmosis. When the osmotic entry of water into the cell or the system, this is called as endoosmosis. What will happen according to the endoosmosis? The cell will swell up. Exo means when the osmotic withdrawal of water from the cell is done, that means the water movement is done from the cell to the surrounding. This is called as exoosmosis. In this case, what will happen? The cell will shrink. Okay, so when we place a cell in high concentration of water, in a higher concentration, the water comes out of the cell by the process of exoosmosis. You can see that when the cell is kept in the hypotonic, that means outside more H2O is there and inside less H2O is there. You can see the diagram. What will happen? The H2O molecules will start going inside the cell, endoosmosis, and the cell is going to swell up. You can see the round or spherical structure. This shows that it has become turgid. The water will start pushing the cell wall. But in this case, you can see that when the cell is kept in the hypertonic, that means inside water is more, outside water is less. What will happen? Exoosmosis will take place and the cell will shrink. You can see that cell has shrunk. So you have understood the meaning of endoosmosis, exoosmosis, endocytosis, exocytosis. Next one, we will do the cell wall. Just to give you a brief of it, what is the meaning of cell wall? Rigid, semi-elastic, semi-transparent and protective covering present outside the plasma membrane in the plant cells, fungus and prokaryotes. Prokaryotes are those cells which are called as primitive cells, in which nucleus is not completely formed. So cell wall is generally found in the plant cell, fungus and the prokaryotes, and they are after the cell membrane. But in each of the organisms, the component of the cell wall is different. In the plants, it is made up of cellulose. In the fungus, is also called as chitin, and sometimes fungal cellulose is also there, which is called as chitin actually. Extra deposit of lignin, suberin and cutin may be present during the secondary thickening. In the plant cells, you must be seeing, like when the plant cells matures, in the cell wall, some kind of deposits of lignin, suberin and cutin takes place. So what happens? It becomes rigid, or basically it becomes dead, cork type of cells, that is called as lignin, suberin and cutin. This deposits in the cell wall after the maturation of the cell in the plants. A cementing layer called as middle lamella is present between the walls of the two cells. Now you can see that if this is one wall of one cell of the plant, this is another. There is one more layer formed in the plant. This is called as middle lamella, which helps in the transportation or any kind of communication between the two adjacent cells. Cell wall possesses small pores through which adjacent cells remains connected, called as plasmodesmata. Now, that is what I say. Middle lamella is there, and the formation of, in the middle lamella, there is a formation of plasmodesmata takes place, which is present in the form of small pores on the cell wall, which helps to connect the neighboring cells, and the transportation of the things or any kind of communication is done between the two adjacent cells with the help of the plasmodesmata, which is basically done by the middle lamella. Okay, so here I end up my class here at this topic. Hope you all have understood this, all the basically the transport system we have done. This is very important. See the video again and again and try to understand. Thank you, children.